DNA replication, books Watson. The chemistry of DNA synthesis, the primer template junction is the first thing that form. Agent structure is being completely described. The DNA is synthesized by extending the 3s end of the primer. The phosphodiester bond is formed in the SN2 reactions. The leaving the group for the reaction is the pyrophosphate. Of the double helix have an antipolar orientation. HTV means the nucleotide triphosphate, monophosphate polymerized pyrophosphate release. The pyrophosphate bond is break to give rise to inorganic phosphate with high energy yield. And this uses the ends of pyrophosphate hydrolysis. The mechanism of DNA polymer. The DNA polymer uses a single active site to catalyze the addition of any 4-deoxyribonucleic triphosphate (DNTP). The DNA polymer monitors the ability of the incoming nucleotide to form an ATGC base pair, rather than detecting the exact nucleotide that enters the active site. The incorrect base pair leads to dramatically lower the rate of the nucleotide addition as a result of catalytic unfavorable alignment of these substances. This is an example of the kinetic proofreading. Indeed, the rate of the incorporation of an incorrect nucleotide is as much as 10,000-fold. The DNA polymer shows an impressive ability to distinguish between the ribonucleotide and deoxyribonucleic triphosphate. The methyl group is not involved in the base pairing. The newly synthesized DNA molecule doesn't have methylated, where the parent cell contains the methylated strand. After some time of replications, the newly synthesized DNA molecules or replicated DNA polymer becomes methylated at the adenine site of a GATC sequence. The nucleotide binding pockets cannot accumulate the 2-deoxyhydroxy on an incoming nucleotide. This space is occupied by the 2 amino acids that mark the van der Waals contact with the sugar ring. This structure reveals that DNA substrate is sited in a large cleft. The ring resembles particularly closed right hand. The three domains of the polymerase are called the thumb, finger, and palm. The palm domain is composed of beta sheets and contains the primary element of the catalytic site. The palm domain is composed of beta sheet and contains the primary elements of the catalytic site. 2 different metal ions, magnesium and zinc. One metal ion reduces the affinity of 3-deoxyhydroxy group of hydrogen. Yes. And second is metal ion is coordinate the negative charge of the beta phosphate and gamma phosphate of the DNTP and stabilize the pyrophosphate. The palm domain also monitors the base pairing of the most recently added nucleotides. The palm, the thumb, the finger. The once acquired base pair is formed between the incoming DNTP and the template. The finger domain moves to the enclosed DNTP. The finger domain also associates with the template region, leading to a nearly 90-degree turn of the phosphodiester backbone between the first and second base of the template. The thumb interacts with the DNA, but as first it maintains the 2 correct positions of the primer. An active site of second thumb helps to maintain the stronger association between the DNA polymer and its substrate. 2 metal ions bound to DNA polymer catalyze the nucleotide additions. Metal ions primarily interact with 3-hydroxy, resulting in reduced association between the O oxygen and the hydrogen. This leaves nucleophilic 3O-. The metal ion B interacts with the triphosphate of the incoming DNTP to neutralize the negative charge. Those events is strongly stimulated by the correct base pairing between the incoming DNTP and the template. DNA polymers are processive enzymes. Thionucleotide per second. Possessive nature of the DNA polymerase possessivity. Degree of processivity, and has given is its initial binding of the polymers to the primer template junction, that is rate limiting for DNA synthesis as it consumes more time than in the replication of the entire DNA molecules. And once the bound nucleotide, it becomes rapid and fast. Structure given is very fast, as told. Structure given. The open binding of the non-processive enzymes are a single DNTP at the 3' end of the primer and then is released from the new primer template junction. In contrast, a processive DNA polymerase adds many DNTP each time it's bind to the template. These interactions include the electrostatic interaction between the phosphate backbone and the thumb domain and interactions. A system based only on a base pair geometry and in complementary between the base cannot reach the extraordinary high levels of accuracy that are observed for the DNA synthesis in the cells. So about 1 in 10 to 5 times, yes. The wrong totem reforms. 6-metcalptopurine, 6-MP are analogs to the nucleotide because it inhibits the synthesis of the pyrimidine to purine nucleotide respectively. Structure of the common commemorative variants of the DNA replications. The proliferating exonuclease. The enzyme degrades the DNA starting from the 3' DNA end. The role for these exonucleases becomes clear when it was determined that they have a strong preference to degree of DNA containing the mismatched base pair. The exonuclease removes these nucleotides from the 3' end of this primer strand. Base pair DNA alters the geometry between the 3' hydroxy and incoming nucleotide base pair for the fluoro interaction within the farm region. When mismatched nucleotide is added, it both decreases the rate of the new nucleotide addition and increases the rate of the proliferating exonuclease activity. The proliferating occurs within the releasing of the DNA from the polymerase when a mismatched base pair is present in the polymerase active site. The primer template junction is destabilized, creating several base pairs unpaired DNA. The exonuclease active site has a tenfold higher affinity for the single-stranded 3' ends. The removal of the mismatched base pair allows the primer template junction to reform and rebind the polymerase active site, enabling the DNA synthesis to continue. Delete K. On average, DNA polymerase is inserted into the incorrect nucleotide for 10 to 5 nucleotide error. This additional level of accuracy is provided by the post-replication mismatch repair process. The junction between the new separated template strand and the unreplicated duplex DNA is known as the replication fork. And very length than the nucleotide organic fragments. All DNA polymer requires primer with 3' hydroxy. They cannot initiate the DNA strand de novo. For new strand, DNA requires RNA primer. The ability of RNA polymerase to do what DNA polymerase can't, the start new RNA polymerase by de novo by using the enzyme primase. And it is a specialized RNA polymerase. Its leading strand requires only a single RNA primer. In contrast, the discontinuous synthesis of the lagging strand means that new primers are needed for organic fragments. Primers refers to the initiate RNA synthesis using a single-stranded DNA template containing a particular trimer, GTA in case of E. coli. Replication fork called DNA helicase. RNA primer must be removed to complete the DNA replication. To replace the RNA primers with DNA, an enzyme called RNase recognizes and removes the most of each RNA primer. The H for hybrid, the RNase removes all the RNA primer except the ribonucleotide directly links to the DNA end. 5' exonuclease that degrades the RNA or DNA forms their 5' end. Removal of the RNA primers from the newly synthesized DNA. The sequence function RNase. The nick in the DNA can be repaired by an enzyme called the DNA ligase. The DNA ligase uses high-energy cofactors such as the ATP to create the phosphodiester bond between the adjacent 5' phosphate and 3' hydroxy group. DNA helicase unwinds the double helix in advance. DNA helicase separates the two strands of the double helix. ATP is added to the DNA helicase bound with a single-stranded DNA. The DNA helicase has 3' 5' 3' polarity. Yes, the DNA helicase has the 5' polarity. Be bound to the lagging strand template. Helicase bound to the lagging strand, not leading strand. By the sixth subunit of the helicase, the single DNA encircled. This subunit has a hairpin protein loop that binds with phosphate of the DNA backbone and its two adjacent ribose components. Single-stranded binding proteins stabilize the single-stranded DNA binding before replication, binding another SSB to the immediately adjacent single-stranded DNA. This is called cooperative binding. Cooperative binding ensures the single-stranded DNA is rapidly encoded by the SSB as it emerges from the DNA helicase. Mainly contact single-stranded DNA through with illustrative interaction with the phosphate backbone and stacking interaction with the DNA base. Limiting amount of SSB is bound to 4 of the 9 SSB DNA molecules shown. It assumes that the more extended conformation is that inhibits the formation of the intermolecular base pair. Topoisomer removes the supercoils produced by the DNA unwinding at the replication fork. The supercoils is introduced by the action of the DNA helicase and removes the topoisomerase. This enzyme does this way by breaking either of the one of the both strands of the DNA without letting go of the DNA passing the same number of DNA strands through the break. Surveillance. Replication fork enzyme extends the range. DNA polymerase can only efficiently extend 3D hydroxy primase. The action of the topo 2 removes the positive supercoils induced by the replication. Positive replication means towards the movement of the DNA. The replication is positive, and on the opposite side is called DNA of negative supercoiling. So this positive supercoiling is removed by the topo isomerase 2. By reducing this side, it's inducing the opposite side. Occur only once every 20 base pair replicators. Type 1 replacement can also remove the positive supercoil generated by the replication fork. Note the supercoils positive in front of the replication fork is shown at the right hand of toroidal with right. The use of the primase is also imposed the requirement for the removal of the RNA primers to complete the replication. It is noteworthy that both the DNA helicase and topoisomerase performs their function without permanently altering the chemical structure of the DNA synthesizing of new molecules. DNA helicase breaks only the hydrogen bonds hold two strands of DNA together without breaking any covalent bond. Although topoisomerase breaks one of the two strands of targeted DNA covalently bonds, each bond broken is precisely reformed before the topoisomerase releases the DNA, and with an altered conformation. The specialization of the DNA polymerase. E. coli has at least 5 DNA polymers, that is DNA polymerase 3 is 3. DNA polymerase 3 is the primary enzyme involved in the replication of the chromosome because of the entire 46 mega-MB E. coli genome replicated in 2 replication fork. This is generally found as part of the rhizotopic complex that confers with very high positivity a complex known as the DNA polymerase 3 holoenzyme. DNA polymerase 1 is specialized for the removal of the RNA primers that are used in initiate the DNA synthesis. The 5' exonuclease that allows the DNA polymerase 1 to remove the RNA or DNA immediately upstream of the site of the DNA synthesis. Sliding clamps. Sliding DNA clamps. Thus, when polymerase completes the replication of the straight DNA, it is released from the sliding clamp. Once released from the DNA, polymer sliding clamps are not immediately removed from the replicated DNA. Instead of their proteins that function side replic DNA synthesis interact with the clamp proteins.